still have a few seats gathered here. If you want to come down and fill them in, a few in the front and a few in between. My name is Andrew Kreitz, and I'm pleased to welcome you to the 2015 Larkin Lecture. Thank you for joining us tonight. I'd like to acknowledge first that we're on the traditional ancestral unceded territory of the Musqueam First Nation. We're gathered this evening to celebrate the legacy of Peter Larkin. For those of you who did not know him, Peter Larkin was recognized in 1988 as a university professor for his contributions to UBC. Now, this is a very special title that has been bestowed on very few UBC faculty members. His citation noted his outstanding and original contributions to the mathematical modeling of fish population dynamics, the theory of resource management, predator-prey relationships, and science policy mechanisms, as well as his outstanding leadership in academic administration. He was also recognized for his contributions to society by the Order of British Columbia and the Order of Canada. Peter Larkin joined UBC in 1948 and simultaneously became the chief fisheries biologist of the BC Game Commission. He took on many leadership roles at UBC, including being the head of the Department of Zoology, the Dean of Graduate Studies, Associate Vice President, and Vice President of Research. Now, in addition to his administrative responsibilities, Peter Larkin also played a significant role in teaching and training of graduate and undergraduate students. As one of his former students, I particularly appreciated and admired his sense of humor, his ability to write papers as well as skits, poems, and songs. They were legendary. And particularly his dedication to research excellence and mentorship. It's a legacy that continues to live on at UBC. I'd like to now call upon Dean of Science, Simon Peacock, to say a few words.
Thank you, Andrew. Dr. Peter Larkin, as we just heard, had a remarkably distinguished career as a teacher, as a fisheries biologist, as an administrative leader, and as a person. I think one of his more important accomplishments was when he founded the UBC Institute of Fisheries back around 1950. And over the years, that institute grew and changed in many ways, and unfortunately it did close, as I understand it, in the late 60s, to be reopened in 1991, focused on interdisciplinary research on fisheries. In the years since, the Fisheries Center grew in reach and in reputation, and in July of this year, the Fisheries Center became the Institute for the Oceans and Fisheries, IOF, with an expanded mandate for collaboration and impact across the disciplines and sectors. In my opinion, the opinion of others, the IOF is urgently needed. The world's oceans are in crisis, and we need solutions. The IOF seeks to understand and address the impacts of our changing global environment on marine and freshwater ecosystems and on ecosystem services. It brings together Canadian and international experts to conduct research and outreach across the natural and social sciences. The IOF is set to lead the way to healthy and sustainable marine and freshwater systems through excellent research, inspirational education, and innovative societal engagement. I'm very grateful for Andrew Kreitz and Billy Christensen, who have agreed to serve as the founding co-directors of the IOF until a permanent director is appointed. The IOF builds upon the outstanding reputation of the Fisheries Center personnel, and we are expanding that to include researchers from across UBC. One of the goals of the IOF is to understand the world's marine systems. In order to do that, researchers must gather and assess vast amounts of data. As I toured the building earlier today, you can see some of the data sets that we have here. An incredible amount of work goes into assembling, compiling those data sets, analyzing those data sets. To assist the IOF in this ambitious task, I'm pleased to announce that we will be devoting a Canada Research Chair in Statistical Ecology with an emphasis on marine research. This chair will be a search conducted jointly by the Department of Statistics and the IOF. And this CRC position has been made possible in part by the generous contribution of the Pacific Salmon Foundation and the leadership of Dr. Brian Waddell. And I'd like to invite Brian to say a few words.
Well, thank you, Simon. This really is a wonderful day to partake in. It surprises me, but when I look back at my calendar, we've been talking about this for over 2 years. And so I want to recognize the university for stick-to-itness. Right? I think this is a great idea, and obviously you guys did as well, and I really do think that there's great opportunity for the IOF in the future. You know, I think that if you were to ask me, one of the questions posed was, why do we support this? Well, since I came out to the West Coast in 1979, I've interacted with a number of the professors here, particularly Carl Walters, but UBC is the natural academic lead in BC. It's great to have the other universities and that, but I think when you look at excellence in leadership, and we clearly think of UBC first. What did I say, by the way? Quoting me? PSF wanted to speak for a second because we have been trying to mount a large ecosystem study in the Strait of Georgia since 2009, and it's not cheap, and so we failed in the short term. But I'm happy to say that we now have an international program valued at $20 million for the next 5 years working internationally, and we really do believe that we need the leadership of a center like the IOF at UBC, and that we will be very pleased to contribute funds to ensure that this proceeds or goes ahead, particularly the chair that you've just announced. I think that we really do need this sort of analytical leadership in the CRC. We clearly know that we need a new Carl Walters, and that, but we'll see Carl in a second. And what I've loved about Carl is that this is a guy who has had an incredible ability to train young people. He has contributed internationally for a long period of time. Many of the people he's trained have actually worked with me at the Department of Fisheries and Oceans over time, and I think that the new chair really is an essential contribution to BC. So I'm very, very pleased to be here tonight and to support the establishment of the IOF and to hear that you're announcing the CRC. And I think that if UBC picks up the challenge of the opportunities that the IO gives, and I hope that you will have learned or listened to putting as much effort into the Strait of Georgia and local BC waters as you do globally, then BC will have a very bright future. So thanks very much.
Thank you, Brian. Another key supporter of the IOF is the Tula Foundation and the Hawkeye Institute. In fact, a number of you here tonight are Hawkeye Fellows. I'd like to invite Eric Peterson, founder of the Tula Foundation and the Hawkeye Institute, to say a few words.
Thank you very much. It's great to be here. It's a little bit timely, I guess. I was, when I graduated, Professor Larkin was the head of zoology, so that kind of dates me. But it's fantastic to be here. I feel with Hawkeye that we really have kind of complementary resources and capability. And I'm being rather presumptuous to say that relative to UBC. But certainly where we're strong is in our field resources and our field stations, our ability to get out there in the field and do research. And it's wonderful to have the partnership with UBC, which is the absolutely phenomenal resources and capability that you have under the roof here. One of the great things about field stations and cruises are, first of all, you get out in the field, which is wonderful. But the second big piece of it is the kind of collaboration that it engenders. You really get to know your collaborators, your co-workers, and it's an amazing scientific experience. And I think that the way that I see IOF, it's a little bit of an opportunity to create a collaborative environment at UBC to draw together all the resources that are here. And so I think it's a fantastic idea. So I would say it's the kind of thing that should lead to collaboration amongst yourselves, collaboration with us, collaboration regionally, and collaboration internationally. And I think that Scientists are like human beings. We're social creatures, and we get the best work done when we work together. So I congratulate and really look forward to IOF. Thank you very much.
Thank you, Eric. Next, I'd like to call upon Gillian Instead, one of Peter Larkin's daughters, to say a few words on behalf of the family.
Thank you, Andrew. So when deciding what I wanted to say to you today, I got doing a little digging. And I was lucky enough to come across a video from the 1992 UBC convocation ceremony, at which my father received an honorary DSc. The short address that he gave to the science graduates spoke to many of the same themes that I'd like to touch upon when addressing the Larkin lecture audience. So I'm going to briefly summarize what he said that day. He started out by talking about how he and my mom graduated with biology degrees from the University of Saskatchewan on a spring day, just like those students in the audience in 1992. He talked about how his head was stuffed full of the latest and greatest knowledge, and he figured that he was set for life. Then a few years later, somebody discovered DNA. He hadn't had any lectures on that. Then computers and software came along. He hadn't had any courses on that. And he continued with similar examples of new findings in geology and chemistry. In his mind, all of these examples served to demonstrate that not only were there new things coming along that he hadn't been taught, but some of the things that he had learned were just plain wrong. So he wrote to the U of S and asked for a refund. Unfortunately, the university didn't agree. They came back saying they couldn't be responsible for post-graduation discoveries. Or words to that effect. He then went on to tell the audience that UBC unfortunately had the same backward policy. He proceeded to then address then-President David Strangway directly, asking for change. He pointed out that the scientific literature was expanding at an astronomical rate. The literature was doubling every 8 years, and in some disciplines, like molecular biology, it was doubling every 18 months. This meant that more than 90% of what would be known by the year 2000, which was only 8 years away, had not yet even been discovered. The figures are likely equally overwhelming today. But he had a solution. He suggested that graduating students should be sent forth into the world with a best before label that indicated when their education expired. In order to be relabeled, students had to come back for refresher courses. Thus, he argued that graduation diplomas should come with detachable coupons for refresher courses, redeemable, say, within 100 years of graduation. All kidding aside, he then delivered his key message. He pointed out that many people in society don't have the opportunity to go to university, and many of those in university don't take science. As science graduates, that class of students was joining less than 2% of the population of Canada, and far, far less than 1% of the population of the world. He maintained it was the societal responsibility of this elite group to ask and answer questions, sort fact from fiction, and reveal truths and falsehoods. In other words, it was their obligation to use their powers of analysis to weigh the evidence and speak up when others sought to fool the public, be it for fame, fortune, or political gain. He firmly believed that when you walk down a career path in science, the skills you take with you from your university education with respect to how to learn will serve to keep you well informed throughout your life. This lecture series meant a lot to my father when it was established, and it continues to be very special to our family. First, it honors and celebrates my father's excellence in this medium as a wonderful way to impart information and engender discussion. Second, it provides the opportunity for members of the Institute for the Oceans and Fisheries community and beyond to interact face-to-face with scientists of international renown. Reading someone's books and papers does not in the least compare with meeting with them directly, engaging with them. Thanks to this lecture series, 11 such experts have come to share their passion for fishery science. So on behalf of my sister Margaret, as well as my other sisters who couldn't join us today, I'd like to thank you all for coming in celebration of lifelong learning. And I thank Dr. Beamish for accepting the invitation to be the 12th Larkin Lecture.
Only one more, and then you'll be
One
up.
more from Peter Larkin, protege. I'm Earl Walters. Peter Larkin hired me in 1969. It's a great honor for me to introduce Dr. Richard Beamish, a great old friend since the early 70s. He grew up in Ontario, got his PhD from the University of Toronto. You can't fault him for that, because he got away. He went to the Freshwater Institute, where he gained worldwide renown for the discovery of acid rain in Canada. Came to BC, to the Pacific Biological Station, and worked there for years, became the director of the station. Became very well known for, in fact, a world leader in the study of regime shifts in fisheries and fisheries oceanography. He has this kind of nasty little secret. When nobody's looking, he sneaks away into his lab, where he has a lab full of nasty sea lampreys, which are this great research love in life. Despite the sea lamprey business, he's been widely honored. He has received the Order of Canada, the Order of British Columbia. He's a fellow of the Royal Society of Canada, and he's received a whole bunch of other awards. So without further ado, we welcome you, Dick. All right,
And thank you for coming.
Now, what am I supposed to do here? Hang on. What is it? Oh, well, I'm being connected. The introductions are overwhelming, because much of what I'm going to say is what you've already heard. I get some water to get that started, and because I will definitely need some. I'm almost getting too old for this, you know. The title is What the Past Tells Us About the Future of Fisheries and Oceans Research in British Columbia. I have some notes. I may need them. I don't know whether I need them or not. But what I wanted to do was, I begin by, a number of years ago, I can't really remember when, but it was when Martha Piper was president. I believe she's currently president for a short time. And she had, let's call it a reception, I believe it was at her residence. And she invited people who she called friends of the University of British Columbia, and I was invited to that. And I am a friend of the University of British Columbia. And I'm telling you that is because I've always had incredible respect for the science, the fishery science that's been done at the University of British Columbia, that's been done here. And this is what Brian Riddle has said to everybody, is that when we heard that, in addition to what you've already been doing, that you are now going to have a focus on fisheries and oceans in British Columbia, this is absolutely spectacular. And so this is what I wanted to say. That you really now are the All Blacks of fisheries science. And hopefully there's no one in the room that doesn't know what this is all about. If not, you watch Saturday morning. Okay? And so just now, what I've done is because I want to talk about the past and then put that into perspective of the future and looking at research opportunities. And so to do that, I've gone back and looked at some of Peter Larkin's talks, and I've extracted some quotes from the talks that would introduce a number of the topics, which I'm calling stories. They are examples, but examples are a little bit too scientific and stories are a lot more friendly, so that's what I call them, all right? I don't know if there's anything else I need to say by way of introduction. I think that's it, so let's get started. And am I doing something wrong? It's quite possible. All right, so this is just a little bit of background. I wasn't really sure who would be here. And so what I've done is to put the value of seafood that's produced in British Columbia and summarized in 1950, 1970, and then in the present. And these are dollar values that are current. And it's important because some of the quotes that I'm using are from the early 1970s, and you can see the importance of salmon then in the 1950 and 1970, and then see how it diminishes in value, but recognizing that the thinking certainly in the past, and by the way, when I refer to the past, I'm usually thinking about 1970, so that's about roughly what the past is for this presentation, okay? And then the various dollar values for the various groups of fishes, but then getting down to cultured salmon and looking at the incredible value of cultured salmon in recent years that was not in the past. And then down at the bottom, a summary of all seafood, which really didn't change much in terms of value between 1950 and 1970. It currently is around, it's almost about double. So that's the background that I think we need to put the rest of this into perspective. So here we go. All right. Story number one, and there's two parts to this, but I want to read this, and I think I can read it looking at it this way. I can't read it from here. Yes, I can. Yes, I can. So this is from Play It Again, Sam. And I'm sure that some of you know that this was an essay that Peter wrote in support of the Salmon Enhancement Program. And here he writes, there seems to be lots of headroom for the expansion of salmon populations before rearing areas of the ocean become a limiting factor. And what's highly relevant about this was that when the Salmon Enhancement Program was eventually presented to Treasury Board, I believe in 1977, at the last minute, the last couple of hours, I'm told that, that government officials decided to pull out the experimental component of it because they were afraid that Treasury Board would not support a program that any experimentation to it, and they wanted to show that this was a proven technology. Now the next quote is from a little bit later, and by the way, the talks that these are from are listed there. I'm not going to bother reading them out. And here he writes, my feel for it is that catches in the whole of the North Pacific today, and this is 1988, represent about 2/3 to 3/4 of the potential catch. And so if you look at the total catch of all salmon by all countries, now I was going to use a pointer, but I think it's probably easier just to avoid pointing to that. You could see that in the early 1970s, when Play It Again, Sam was written, the total catches were around 400,000 tons, and then in the late 80s, around 600,000 tons. And then the record catch is broken in 1995, broken again in 2007, and broken again in 2009, and then recently the record catch was in 2013 of 1.1 million metric tons of catch. So going back to the quote in the late 1980s, Peter Larkin wasn't too far off what he projected the catch would be. But what was not expected was this. And this is the red line is the percentage of the total catch by Canada. And the percentage there well, it was in the early 70s, up around 15, even 20%, maybe a little bit higher. And then staying somewhere around 15% into the early 1980s, and then declining dramatically, really, down to just a couple percent. And so the issue here is that in the past there was this belief expressed by Peter Larkin, but that was a common belief by many people, and that changes. And it changes in part because of this. It changes because pink salmon increase in abundance. This is the all-nation catch of pink salmon through to the present. And it changes because the industry in British Columbia, certainly beginning in the 1990s, really no longer values the catch of pink salmon. But what I think stands out in what you're looking at there are in the last 10 or 15 years, you can see some on and off year changes, which I'm going to talk about in a minute. So again, going back in time a little bit, I think the first investigators to really detect that there was a change in pink salmon production by Godfrey, and then a nice paper by Bill Ricker in 1978 pointing out that the average sizes of odd year and even year pink salmon differ. And then the geneticists get hold of this, and the two labs, which I'll just call the Siebes and the Beachams, who do excellent work, even though they are a little competitive, and maybe it's more than that. And both these labs publish papers that show that there is a pretty, I think, distinct genetic difference between odd and even year pink salmon. And they write that there is 5.5 times more genetic diversity between brood lines than within the brood lines. And so going back to the total catch then of odd year and even year pink salmon, this time separating the catch by odd and even years, there's a clear difference in the total catch, with the even years being blue and the odd years being red. And if you look at it this way, which is a deviation from 1990 to the present, with the means there, with the odd year being on top and the even year on the bottom, and those differences are significant. So there is a significant difference in the production of odd and even year pink salmon, but looking at the top, at the odd year trends, there's a pretty clear increasing trend in odd year production, while even year pink production, which is on the bottom, is more or less random, not quite, but still a very different trend in odd and even year pink salmon. And we're beginning to see some research that's indicating what the explanation might be. And here is a master's study just completed by Melissa, I'm going to pronounce her name, Prechtel, and Megan McPhee, who Megan is a professor up at the University of Fairbanks. And this is a master's study that's just in the process of being published as a primary paper. And they were able to show, looking at odd and even year pink salmon, that the odd year pink salmon had a significantly greater level of IGF-1, which is an insulin-like growth factor, which is an index of growth. And then the second point, which is that the odd year pink salmon also have a significantly lower energy density. So the summary then, which they express as a hypothesis, and you can read that, and the quotation marks are mine, but the interpretation is essentially that there is a genetic difference between odd and year pink salmon, and a genetic basis for the differences in production of odd and even year pink salmon in the last 20-25 years. And the difference may be that the odd year pink salmon use energy differently, and they basically bet on finding food during the winter, which may be happening because of essentially a global warming change that produces more food in the winter than in the past. And isn't that a tremendous opportunity for you guys to do some research? Story number one. Okay, story number two. Again, a quote from Play It Again, Sam. And any program of salmon enhancement should be coupled by a program of experimentation and evaluation. So it's a little bit like what I've already said. And then another quote, this time a little bit later, and this time writing that, and this is again emphasis on research, but it's important to remember that it is the freshwater environment that at present limits the total production of salmon. And while there are variations in marine survival, as a general rule, the greater the total output of young salmon to the sea, the greater is the eventual return of adults. And this, I think, was a common belief, acceptance, if you want, of scientists right up into the late 1980s. And I'm going to show you some examples that indicate that both interpretations that the number of juveniles is a legitimate interpretation, but that interpretation probably started to change in the early 1980s. And this is something that I think some of you are familiar, certainly Carl Walters and Brian Riddle. And this is just a quick example to show you that there was this dramatic decline in the production of Chinook salmon in the Strait of Georgia, with a catch of, 800,000 individuals in the late 1970s, and then declining rather dramatically in the 1980s, and then I think the catch now is around 20,000 individuals. So a catch in the 1980s that was quite exceptional, and with the thinking that you could even increase, that's where Play It Again, Sam, I think, related to, and then the possibility that the enhancement program could even increase on this, but that didn't happen. And I'm going to, from time to time, mention that what is happening in the 1980s that really was not appreciated by all of us was what I'm just going to call an undetected change in the ocean environment. And we still don't know what that change is. I don't think there's any question that there is a change, and I'll show you some examples of that in a way affects marine survival. And that wasn't anticipated in the past, and we gradually learned to appreciate that was a major influence. So just another slide to show you the marine survival of juvenile Chinook salmon. This is in the Strait of Georgia and Puget Sound, just to demonstrate the synchrony, but I just wanted to quickly show you the marine survival of Cowichan hatchery Chinook salmon, again showing let's call it a rather abrupt decline in marine survival beginning around the 1990s. And again, there is a repeated, I think, evidence of something happening in the 1990s that affects the trends of the production of a number of species. But again, this is what I'm calling an undetected change in the ocean environment, in the climate, that affects the overall production. And then one more example of marine survival, specifically looking at the Cowichan River, Cowichan hatchery, Chinook salmon. And part of this is based on a study that we did in 2008. And this is to show you the tremendous mortality that occurs in that first ocean year. Now, we benefited in 2008 by knowing that the juvenile Cowichan Chinook salmon mostly stay in the Strait of Georgia. We were able to determine that through our surveys and through DNA. And that allowed us to make swept volume estimates, and with a fairly large mortality or survival through to early July, and then by October, about just less than 1% had survived through to October. But because all of the juveniles were code wire tagged that year, when the brood year was finally over with and the adults had returned, the marine survival was less than 1%. And so the point is that this was really unexpected in the past, and then we had this substantial change, which more or less is still occurring with some small changes. But it's this next example that I wanted to focus on, and that's this. And I think it's time to have a drink of water. Well, my voice won't last for a whole hour, so... Okay. So this is, I think, I'll try not to use the word remarkable too many times, but I think this is quite remarkable. So we all understand the decline that we've seen in Chinook salmon, and it's even into Alaska. But here we're looking at an aggregate of Chinook salmon, South Thompson Chinook salmon, showing that at about the time we started to see the decline in the Cowichan Chinook salmon, you're starting to see an increase in production of these populations in South Thompson. And remember that the point of these stories is that I'm suggesting that this is different than what had happened in the past, and these are opportunities for research in the future. And so, looking at the ocean entry time of South Thompson Chinook salmon smolts, and these are data from Chris Neville and her colleagues, and this is rotary screwed trap catches, and with the DNA to show that the South Thompson Chinook salmon are entering the Strait of Georgia substantially later than all other juveniles. And what I'm proposing is they are using the Strait of Georgia environment differently than the majority of earlier entering Chinook salmon. And then these are some catches that we made in our trawl surveys. The top survey is a little bit later in July, so the dark bars are the sizes of the South Thompson Chinook salmon, and they are considerably smaller. And then the second one, just another example to show that the South Thompson Chinook salmon that you already saw enter later, and they are also smaller. And then another example, this time with DNA, with a nice pretty blue color, showing the South Thompson Chinook salmon abundance, and then but showing in July very little, very few South Thompson Chinook in the Strait of Georgia. But by September, high percentage of the juveniles are South Thompson. And then one more slide that shows these are swept volume abundance estimates. And so if you look at this, looking at abundance estimates in July and in September, and I just used 2 years, and what you see over on the far right is that there is a replacement of the earlier entering Chinook smolts, juveniles, which are the majority of the population, with the South Thompson fish. So by September, a large majority of the juvenile Chinook salmon in the Strait of Georgia are from South Thompson. Now, I'm not going to talk too much about this, but we did an acoustic tagging survey, and for some of you that aren't familiar with acoustic tagging, the tag that's implanted in the juvenile salmon will be hopefully detected by underwater listening lines that are in the northern part of Queen Charlotte Strait and Juan de Fuca Strait. So in theory, and mostly in practice, a fish with an acoustic tag, when it leaves the Strait of Georgia, will be detected. But what we found, without getting into the problems of interpreting acoustic tags, what we found is that very few Chinook salmon left the Strait of Georgia. And that's not a bad sample size. And of the fish that did leave, the South Thompson fish, there was more of them. So when you go back to that earlier slide where you had the abundance estimates, And then there was this replacement of the earlier entering and the majority of Chinook salmon smolts by the South Thompson fish. We really had no record of those earlier those earlier Chinook salmon actually leaving the Strait of Georgia, with the possibility that probably most of them died. And again, what's highly relevant is that what was expected in the past was not what was happening, but this was happening. And there's a bit of a problem with this, that if Carl Walters, well, it won't take him too many minutes to figure out what the problem is, but in general, what this is, it's not too bad. And what I'm showing is that there is a relationship between the escapement and the subsequent production. This goes back in time. And why I'm showing you this is really for this reason here, is that what Peter Larkin wrote in the late 1980s and what a majority of people thought, I think applies legitimate to the South Thompson fish. And it is the number of juveniles that are entering the Strait of Georgia that is important in terms of the return. And so that thinking that we saw in the past, and I'm speculating of course, but I'm speculating that it was perfectly legitimate. That was what the ocean was like, let's say in the 60s and 70s. That the number of juveniles entering the ocean was related to the production. But what happens is you get this undetected change, and it affects the earlier entering Chinook salmon, but the environment that the South Thompson fish are appreciating is the type of environment that the Chinook salmon experienced in the past, all right? All you need to do is figure out exactly what the change was. Okay, now this is part 2 of the same story with a similar quote, but there's a good body of evidence to support the notion that for fish populations, year class size is determined early in life, and environmental conditions set a stage for which density-dependent factors act to regulate numbers. So similar to some of the things that I've already said, and then one more quote, which is that there is a need for long-term, multidisciplinary research program to improve understanding of the ways in which species interact. And a particular need is for studies of recruitment directed at larval ecology and the interrelations of species in nursery areas. All right. And so here's the issue, is that this is what you have now proposed to do. And as we've heard from Peacock Institute, this is work that's being done there, work that's being done in Brian Riddle's program, or whatever it's called. And there is a reasonable amount of research being done on these two topics, as there should be. But I want to present something slightly different, all right? And this is what I want to talk about, this. And that is that we tend to focus on this big picture, trying to interpret what's going on. And I'm suggesting that there is another way of doing it, in addition to what we're already doing. And that's that looking at the smaller, much smaller numbers of populations that are doing really well. And I suppose I would like to say that Tony Farrell reminded me of the Krog principle. But I think, in all fairness, I didn't have a clue of the Krog principle. I'd like to ask if there's how many people know about the Krog principle. That good, because I'd never really heard of it before. But once I had this discussion with Tony, like 6 or 8 months ago, and there's no question that it's highly relevant to what we are doing. And so simply stated here, and it's much shorter than, well, it's roughly what was written a number of years ago. And the Krog principle states that the selection of a particular animal greatly facilitates the understanding of a particular mechanism. And I'm going to show you an example of how I think the Krog principle could be applied not just to sockeye salmon, which is the example, but I think it relates to all salmon. And this is the Harrison C-type sockeye salmon. And I know that some of you are aware of what's going on, but C-type sockeye salmon actually behave like pink salmon. And C-type sockeye salmon enter the Strait of Georgia in the year that they emerge from the gravel. And as most of you know, lake-type sockeye salmon, which are the dominant group, they will spend a year or two in a lake before entering the Strait of Georgia, before entering the ocean. So what has happened, which I said I wouldn't use the term spectacular too many times, but it is spectacular. This is the return of, total return of Harrison sockeye salmon to the Fraser in the last 10 or so years, with estimates, in some cases, over 20%. The red is supposed to be what's the forecast for this year, and I really have no idea what the return was, but it was forecasted to be, I think, 21%. So there is something that is quite remarkable about the survival of Harrison River sockeye, C-type sockeye salmon. This is not much of a graph, but it does show you the residence time of the lake type in the Strait of Georgia, showing that they begin to enter the Strait of Georgia about mid-May and then peak about early June. But here's the point, is that by mid-July, virtually all of the lake type sockeye have left the Strait of Georgia. And looking at DNA for the earlier part of the Strait of Georgia before they leave, that the red being the south, being Harrison, there's very few Harrison in the Strait of Georgia when there are the lake type. And then the abundance of Harrison C-type in the Strait of Georgia in recent years, in September and right through to November. So this is a unique behavior in terms of what juvenile sockeye are doing. And I know that most of you that have studied sockeye salmon in general know that the juvenile sockeye salmon leave the Strait of Georgia, and people used to write, quickly leave the Strait of Georgia. There's some validity to that. But it wasn't completely true. There was always a small number of the C-type sockeye that remained in the Strait of Georgia, and now there are a large number. I just want to point out that they are smaller. They do move into Howe Sound, but just to show that, again, these fish are much smaller when they enter the Strait of Georgia, so they're not surviving because they're bigger, they're surviving because of their entry time. But this is what I want to show. To get to this, this is the catch per unit of effort. This is a relative abundance of the Harrison C-type sockeye salmon in the Strait of Georgia during the period that we are seeing these large returns. And then if you look at the total returns, and this time it's a little more scientifically based because it includes total production. So these are the catch of adults as well as the total returns. There's two brood years, showing that most likely the reason for the increased production is because of something that's going on in the Strait of Georgia. Now wait till you see this. So from diet, without getting into all of the details, but showing that the dominant organism in the diet of the juvenile salmon in September are amphipods. And then just working their way through this slide, the amphipods that are dominant are hyperid amphipods, and one particular species of hyperid amphipods, and I'm never really sure how to pronounce that, but Amphipods is the dominant amphipod in the diet. And looking at, this is a study published by Mackes and his colleagues, excellent summary of of the zooplankton in the Strait of Georgia, but basically showing that this one particular species of hyperid amphipod is the dominant food item of the South Thompson, of the Harrison sockeye salmon. And so something has happened, something has changed in the Strait of Georgia. I'm speculating, of course. Something has changed to result in a change in the dominance of this one particular hyperid amphipod, which I'm told has a lot of lipids in it, and I'm guessing that this is largely the reason that the Harrison sockeye salmon are doing so well. All right? Again, some undetected and clearly unstudied change going on in the Strait of Georgia, which I'm using as an example of you wouldn't have expected this in the past, and now there is something that I think needs to be studied in the future. Okay, the next item. Now, this isn't the most comfortable topic to talk about, all right? But this is something that we do need to talk about. And this is, I think, another insightful comment from Peter Larkin. And I'll read this. There can be little doubt that aquaculture is here to stay, and little doubt that it has a bright future, at least for salmon on the Pacific coast. But you had better believe that the next 20 years will not only see some phenomenal growth in aquaculture production, but also some amazing development of new strains of seafood with greater appeal. And I'm sure that those of you that have listened to Peter Larkin's talks, when he said you had better believe, and this is clearly what has happened. So just to remind you of that earlier slide, to show you that cultured salmon, which is largely farm salmon, has been the reason for this substantial increase in seafood production in British Columbia, with the potential of maybe even doubling it. So, a number of years ago, I don't know, I always say a number of years ago, I really have no idea, but probably 15 years ago. And, I gave a talk, and it was called Getting the Message Out After It's Too Late. And since Brian Riddle is here, I think I will also comment that he and I wrote I think we call it a white paper in the late 1980s outlining what the Department of Fisheries and Oceans should do to basically implement salmon farming. And needless to say, no one really paid any attention to it, and neither Riddle nor I can even find a copy of it. And what I'm showing you here is that about 15 years ago, these were the advisory committees for DFO. And you cannot find a committee there on aquaculture or on salmon farming. And I'm going to suggest that one of the major difficulties that we have had is because that Department of Fisheries and Oceans did not take salmon farming seriously. But that has changed. And largely, not largely, entirely because of the environmental community, and because of the opposition and concern to salmon farming, DFO a few years ago created this aquaculture management group. And this is a group of 50 to 60 employees with these areas of responsibility. So I'm going to suggest that DFO has now taken the issue of aquaculture and salmon farming specifically seriously. But I've got two examples of what has happened in the past. And they are naturally controversial, and I'm not going to spend too much time on them. But some of you may know this particular example. And first of all, look, it's perfectly legitimate for science to produce this paper. And this represents scientists who strongly believed in their analyses, who strongly believed that there was this serious problem. They wrote a paper that passed a peer review process in Science magazine, and it was published. And of course, I'm using it because it basically made headlines all over the world. And this is, I think, perfectly legitimate for science to behave this way. Now, I'll tell you that a few of us wrote a rebuttal to this paper, which is also perfectly legitimate, and it was also published in Science magazine. So this is exactly the way science should work. However, there's some fairly harsh language there. If outbreaks continue, then local extinction is certain, and a 99% collapse in pink salmon abundance is expected in 4 salmon generations. And that was 2006 to 2014. And just so that you know what the production has actually turned out to be, looking at 2014, now Glendale has spawning channels, but look at Capweekon, your wild stocks, looking at a 10-fold increase in production in that 10 years of time. And then one other example, very brief example. This is a highly relevant document. This is a document produced by a report, if you want an understanding, Senate Committee on Fisheries and Oceans. And I believe that they spent about 2 years getting testimony and producing this report that came out at the end of July. And if you haven't read it, I think it's highly relevant that you look at this report. And I just have one quote from that, because another issue that found its way all around the world was this statement that ISA, which is the virus, was found in salmon in British Columbia. And then the Canadian Food Inspection Agency, I believe, looked at about 10,000 individuals, and they did not detect ISA at all. That's all I'm going to say about this, other than this. And that the conclusions is that, as I've already said, salmon farming is now the most important seafood industry in British Columbia. And now, with the valuation of the loonie, it's no longer true, but a year ago, the value of the salmon farming industry in British Columbia was about the same as the catch in Alaska, and as I said, worth more than all seafood produced in British Columbia combined. And I'm saying it was a rough start, but it appears that the industry, and in that talk that I gave with the title, Getting a Message Out After It's Too Late, I said, what did the industry think when they started raising Atlantic salmon in British Columbia? Did they think they could bring Atlantic salmon into British Columbia that produced green peas with all of the issues in the Atlantic without assuring British Columbians that there wasn't any harm? And then the final statement is that I think that it is fair to say that the industry is probably sustainable, and I think that we now have evidence that indicates that they are probably not doing substantial, or not doing harm to wild salmon, but certainly the industry has a way to go socially. Well, that's the most uncomfortable part of this presentation. Okay, this I think is huge, and we've already had some of the introductory comments made about this. And, you know, I really like this quote. There are many scientists who now believe, this is the late 1980s, and while I'm reading this, look at that's the next slide. There are many scientists who now believe that the world is warming up, and there is the suspicion that the pattern of circulation may be changing in the North Pacific. Okay, but look at this. If so, it's bad news for marine survival of British Columbia salmon in general, and Chinook in particular. And that's, you know, some of the earlier quotes. This is just a little bit later, and Peter and scientists are just beginning to see the threat of ocean warming, global warming. And so, look at this. I know some of you know this, but this is the warming of the Strait of Georgia from the play of the game Sam era through to almost the present. Top to bottom, and this is the summary of that slide. And this is a very nice paper published by Salmon and Cummings with the clear statement that the sea surface temperatures warmed by 1.4 degrees from 1979 to 2005, and then the scientific measurement, which is not all that important right now. But this is, again, highly relevant. Increases are related to the upper ocean temperatures throughout the North Pacific, with a 4-month lag showing that large-scale climate events affect the oceanography of the Strait of Georgia. And there really isn't a person, I think, in this room that doesn't appreciate what can happen if those trends that we saw in the 1970s through to the present. Now, there was that cooling, but I think most of us believe that that's a temporary cooling. Now, it could last longer. I have some Russian colleagues that think it will last longer. But even if it lasts a little bit longer, I think that most of us accept that we're going to see that continuation of warming over the next 25 to 35 years. And that's huge, and that's why, again, the decision that you've made here is highly relevant to the future of what's happens in British Columbia. And then the next change. Now, obviously, I think none of us really, it's interesting because if we thought about it, probably would have figured that this was a possibility. But the ocean acidification, which I think is already, well, we've already now accept, and this is a nice messy slide that's all Jeff Marley's fault, all right? And he's in the back and you can blame him for this. And this is now published, but showing this decline in pH. What is remarkable about this is the Vancouver Aquarium that first tells us that we're starting to see the acidification of the Strait of Georgia, a decline in pH. This is a summary from what he and his colleagues have published, and with an indication of up to the mid-1970s of 8.3 to 7.8, and I'm sure that most of you are aware of what pH means, so increasing acidity. But look at this, by the year 2000s, the pH ranged from 7.9 to 7.3, with an extreme low at 7.1 in July, August of 2001. And clearly, there is something that's going on that is of extreme importance. And I thought I would just briefly summarize what has been going on in island scallops. Now, again, maybe not all of you know about island scallops, but it started in 1989, and initially, island scallops bred the Japanese scallop, and then they had some problems, and they, with a parasite, they crossed it with the weather vane, and the scallop that they produced for years, they just called the Pacific scallop. But here's the issue. That starting in 2010, with a low pH, and then this high partial pressure of 750 parts per million, and look, I just threw that in 2013, they had a high partial pressure of 1,400 parts per million. All right, so what, 350 parts per million, without getting into discussion about it, could be about average. So we're seeing huge increases in some situations between partial pressure of CO2 as well as this acidification. This all made the popular press around 2013, and what happened that year was they had 10 million scallops already to market. They were 2 brood years. They wanted to wait until the scallops had spawned so that the meat would be in better conditions. But by August, all of the scholars were dead. And now, all of a sudden, was really when I think we all started to realize that there was this major issue. And it's not just acidification. It's acidification and it's partial pressure of CO2, and it's really that we do not know what's going on, that something is happening. And then I just noted that in 2014, I think it's fair to say that the oyster growers in the Strait of Georgia, although they kept this pretty quiet, they then really started to experience the problem of seed survival. So, a huge issue for the future that, again, is a nice challenge for you guys. And then, my final story. And here Peter Larkin wrote, and we've already heard some of the comments that he's made about the involvement of the public. And a role of science is to provoke public discussion. And here's a quote that I like. I really, actually, I have this, even though I've been retired for a number of years, I stick this up in front of my desk at the biological station, which my wife accuses me of going there every day, but have some coffee and talk to people and see what Sandy McFarland's up to. Anyhow, this is Richard Feynman, who some of you know was a Nobel Prize winning physicist. And he wrote in his book, Surely You're Joking, Mr. Feynman, and he wrote, "Honesty in science is telling intelligent people what they need to know so that they can make intelligent decisions." And what's so highly relevant about this is here's this world-renowned scientist who is saying that, yes, my science, of course, is important, but my responsibility is to ensure that I inform the public about what science really is all about, what science means to them. And a group of us produced this book. I'm not showing you this to promote the book. The book is doing quite well, no problem. All right? The book has a number of chapters. There's 3 chapters. One is by Rick Thompson. And if there's any of you that are trying to understand how the physical oceanography works in the Strait of Georgia, I assure you that it was reading Rick Thompson's chapter that I really understood what was going on. It's superb. All the chapters are written for the general public. There's no citations. There is some technical language from time to time, and it's explained. Another chapter is by Trites, and as you can expect, it's the most popular chapter because everyone likes marine mammals, right? And then just my third example is Terry Glavin, and Terry Glavin writes about the First Peoples, about the First Nations. And this is the best written chapter, but it should be. He's a professional writer. Okay, here's the point. I'll talk about this, but I just want to talk in general terms. Like other scientists in this room, and I publish lots of scientific papers, and we get promoted on our papers, we publish in good journals because we think it's highly relevant, we get a higher citation index, and we think impact factors are important, and I'm sure universities put lots of recognition on the number and the quality of scientific papers. And you know, after all the years of doing that, one of the things, maybe the most important thing about producing this book on the Strait of Georgia, which we just did because we thought it was the right thing to do. And by the way, Riddle was a major sponsor of the book and the Salmon Foundation. Anyhow, what I learned from that book was, you know, we really have done, and I'm going to say, a very poor job informing the general public. And you'll see my last comment here, I'll get to Peter Larkin in a minute, but we do a poor job. And it's now things have changed a little bit with the election, right? If the election had gone the other way, I would have had a lot more things to say. But we do have to to do a much better job, as Richard Feynman said and as Peter Larkin said. You have to communicate objectively to the general public. We have to find a way of passing on that information that we know. What we know, what we don't know, what we need to know. And I don't want to exaggerate too much, but I'm inclined to think now that's almost as important as the scientific literature that we produce. Now I know that it's probably too extreme, but it's pretty close to being what we should do. So the absolute best person that I know of who could communicate science to the industry and to governments, and who had this seamless interface between the general public and science, was Peter Larkin. And I don't think there's any dispute about the ability that he had. Now, I doubt whether we're ever going to find another one that had that ability. But I am saying that this is something that the scientific community has to work really hard to do, is that we need to find a way of communicating the science that we do to the general public objectively. Okay, I'm at the end. Just 2 more slides. So, to summarize then, what I sort of decided, what I thought that our history has told us about the future of fisheries and oceans research. And the first one is that ocean and climate variability change relationships. And I've shown you some examples where what we were thinking in the past, where the number of juveniles entering the ocean was important and basically regulated return, most likely was correct. But something changes. There is this undetectable change that has an impact, and I showed you the marine survival declines, and I think that's legitimate. And then I showed you some examples of some populations that are actually doing well. Again, an indication that there are these trends in the ocean environment, in climate, that have major impacts, and we really don't understand them. And I'm suggesting that, ultimately, we will be forecasting everything reasonably well. But we are a long way from being there, despite Walters and other people here. But we had a good start. The students are far better than anything that I've ever seen, but we still need to do the work that provides the information on the mechanisms. And as we get that, we watch how good the forecasts are. The research is difficult. Look, if we've learned anything in the last 30 or 40 years, we've learned, I think it's fair to say how little we really know because of some of these changes. And I'm saying that the research is difficult because it's multidisciplinary. And boy, I've never seen anything like it that we have now. It's almost too bad that I'm not younger and get back involved again. But look, you've got the Hackeye Institute, you've got the Salmon Foundation, you've got a new government, you've got your UBC, and then you've got other universities that are involved. You've got an incredible opportunity at exactly the right time to begin to work as an integrated team. Expect the unexpected. Do you have anyone other than Riddle who knows who said that? Bill Ricker. Bill Ricker, that's what he said. If we learned anything about Pacific salmons, to expect the unexpected. And then, finally, keep the public informed, but avoid advocacy. And so my final slide is this, is that just like I started by saying, you are now the All Blacks of fisheries science. I really, really believe that your new organization can provide the information that's needed to make wise stewardship decisions in what is going to be the most challenging and important period for fisheries and oceans research, I think, in the history of British Columbia. And that's it. Thank you.
The pink salmon and how the population increased, whereas the Acoustic et al. Paper speculated that the population would crash. Is that Was your reflections on that based on any before or after control impact studies? Because my understanding is that salmon aquaculture management changed largely due to the discussions that occurred after those scientific papers came out, and there was a lot more control of the lice. I just think it's a little bit of an unfair characterization to say that these models in this science paper was complete advocacy and scientifically invalid, which could be one way of interpreting how you presented that data.
Well, I agree with you, and actually I wrote a couple of papers on that. All right, so I do. But, I mean, I'm not trying to be unfair. I'm trying to identify that the science is legitimate, all right? But there is a fair amount of science now. And what I'm trying to comment on is that years ago we didn't have that science. We now have it, all right? And that's my point. I'm definitely not trying to be unfair.
Any other questions? No. So first off, I just want to thank Dr. Beamish for a very stimulating talk, with many points and things for us to think about. I'd also like to present you with a gift, and maybe on the left, and for those that are used to our typical gifts, no, it is not a coffee mug. This is the new issue for the Oceans and Fisheries.
Well, tell me what it is.
Well, you're going to see it in a minute.
Oh, all right.
Anyway, I do want to thank you very much, as well as I'd also like to thank some of the other people who are here with us this evening that helped make this event so successful. Pamela Rosenbaum, who organized the event, Will Schwartz and Andres Cisneros, who organized the open house before the lecture, and of course the Larkin family, who contributing to this event since its inception. And we really appreciate and thank Jillian and Margaret for being here with us today to represent. So thank all of you. Those of you that would like to continue the discussion with Dick, he's going to be here tomorrow morning for a question and answer session. All the students we're hoping will be there. It's going to be in room 107 down here at the end of the lobby area. And at the same time, we also encourage others who would like to come and join in and talk more about some of the ideas that were presented today. Finally, I'd like to give you all a heads up that there's going to be a special IOF event at the end of May, this coming May, 2016. We aim to have an even bigger and better open house, and there'll also be a panel discussion on oceans and fisheries. So I'm hoping you'll all be able to join us then, and since you're now all on our email list, you will hear about that. And finally, I'd like to welcome you and to invite you and everyone here to come out and join us in the atrium for reception. Thank you all.